Hi, this is Miss Litton, and this is a Friday morning with my wonderful Period 3 class. Say hi. Hi. All right, so we're just going to do a quick review of what we discussed so far, and today's notes are just kind of getting the details of something you already learned in our last presentation. Oh, you can't see it? My bad. Here it is. Okay, so when you take the DNA and you use that DNA to make RNA, what is that process called? What? Using DNA to make RNA. What's that process called? Transcription. Good. When you use that RNA that you made in order to build a protein, that's called translation. Okay? We have 3 words you need to know cold. I cannot overestimate the importance, that would be a better way. Overestimate the importance of these 3, differentiating between them. Okay, in your central dogma, when you have DNA and you're making more DNA, when would you be doing that? Why would you ever need to make and copy your entire genome of that cell, all of the DNA? As stage of interphase, because you're getting ready to do mitosis or meiosis. You're making a whole new cell. So if we look at this textbook and consider this textbook to be all the DNA, you're going to copy all of it if you're going to need to have 2 classrooms to put it in. Yes? Okay, now, sometimes I need to build a protein. So I am going to use, I'm not going to copy all of the RNA into an mRNA, just that particular gene. So when you open up a piece of the DNA and uncoil it so it's available to copy, and you make that RNA, it could be one of 3 kinds of RNA. What are the 3 kinds of RNA? Do you remember? Okay. And then, oldest bio buddy, why don't you specify? Oh, probably want a cracker. Hang on 1 second. Here we go. Oldest bio buddy, you differentiate between the 3 kinds of RNA. Go. All right. All of these RNAs were transcribed from the DNA. Yes? All 3 of these RNAs were transcribed from the DNA. Messenger RNA, though, this messenger RNA, it's the one that has the code on it. Remember, every 3 bases on it is called the codon. Okay? The tRNA, it's just a mule. Okay? It's a mule, and what is it carrying for us? It does have an anti-codon on it, but what's the transfer? What is it transferring? Amino acids. It's transferring an amino acid. And then ribosomal RNA, you need several different RNAs to make 3 exactly, to make up the large ribosomal subunit, and 1 kind of RNA to make up the small ribosomal subunit, and some proteins. And the rRNA is going to be the what for protein synthesis? Workbench. Perfect. Okay. So, coming back here. Okay. So when you do transcription, you could make any one of the 3 RNAs, but it is the messenger RNA that you will be translating into a protein. And this is when we talked about when I go from hello to hola, right? I'm in a different language. I just left the nucleic acid language, and I've just gone into the protein. Went from a nucleotide language into an amino acid, right? I've changed. I'm into another one of my 4 important organic molecules, yes? Okay, and it's these proteins that could be structures, muscle, right? Or it could be an enzyme, okay, that I need in order to run the body. Your genetic code in action, your genotype becoming phenotype, is in the process of translation. What has to happen before translation? Transcription. Okay, where you get the information from the DNA. Okay, that's the big picture. Ask your bio buddy, Shadowlands or Sunshine on the big picture? Clarifying questions. Were you guys raising your hands, Justin? Oh, okay. All right. So, when we're here, okay, we can see our DNA up here on top, and the DNA consists of a leading and a lagging strand. And when we did DNA replication, we had to copy both of those, yes? But when we make mRNA, we only need to use the leading strand. Why is that? Because RNA is single-stranded. How else is RNA different? Okay, so wherever you would want to make a complementary pairing, normally adenine would bind with thymine, right? Instead, you would have a nucleotide that has what in it? Uracil. How else is it different? Sugar. Yeah, what sugar is in the RNA? In the RNA, ribose, and in the DNA, it's deoxyribose. Perfect. Okay, so now you make this RNA. Now you make this RNA, and here, this is called mature RNA. And the reason why it's mature RNA is you put a cap, that's a cap, on one end, and then a tail of about 100 to 200 adenines on this end to protect it. And you cut out what's called the introns, which is annoying, and you leave the exons, okay? There are reasons for each, and I'll go into the details of that in just a little bit, okay? But you cut out parts of it and you leave some, okay? Once it's mature, then it can leave through a nuclear pore, and this mRNA is going to go out and get on the workbench. Who's the workbench? Ribosomes, and ribosomes are made out of what kind of RNA? RNA and proteins, sorry. Okay, so the mRNA goes out, here it is, and every 3 bases on that mRNA is called a what? Codon. That will be complementary to a what? Anticodon located on the tRNA. So this tRNA right here is GCU, if that's the tRNA. It could only bind onto the mRNA where it says what? On the G with C, C with G, U with A. Good job. Okay. Who told this mRNA to be GCA right there? The DNA. So the DNA determined the mRNA determines the tRNA, and the tRNA brings the amino acids. So ultimately, it's the DNA determining the sequence of the amino acids in every protein. Yes? Okay, check with BioBuddy. Shadowlands or Sunshine? Go. Okay, now let's talk about things we know. The things we know, okay? So we know one thing for sure. The mRNA, the codons, we know what amino acids are going to get brought to us via the mule, via the tRNA. And we looked at that, we said Nuremberg and Mattei, okay? These two scientists discovered the genetic code. Now, if you look at this, there are 64 boxes in your genetic code. If you add those up. And that's because you have how many bases? How many bases do you have? 4 bases, but they can be in any one of 3 spots, each of those 4 bases. So if you do all the different combinations, you have 64 options there. Okay? Now, you can look up here. Some of these options, there is no amino acid. What do you see there in its place? Stop. Okay? These right here, it's just like stop. Okay? Don't go any farther. These are periods at the end of your 3-word sentences. Okay, let me try again. All right, so you start hooking amino acids, building a protein, right? When do you stop? When you reach a stop codon. It's built, done, go away, you're through. Okay, that's a stop codon. Now I'm going to show you the details of that when we get to the details of translation. I'm just trying to throw it in there for now. Okay, so some of these you're going to see, for instance, if you look at arginine is C. Oh, I want to be yellow. When you look at arginine, okay, you can get arginine this way: CGA, CGC, CGU, CGG. Do you see any more arginine up there? couple other ways to make arginine. So there's multiple ways that you can get that same amino acid brought. So it doesn't matter if your tRNA says CGU. In fact, all that matters is it says C what? G. And down here, you actually could say CG or AG, and either CG, AG, yeah, and either way, you're going to get arginine. See how that works? There's multiple ways to get that one amino acid. How many different amino acids are there? 20. And we have 64 minus 3 stop codons. What's 64 minus 3? 61. We have 61 combinations up here, but we only need 20 different amino acids. Okay? All right. Now, we know that that genetic code is universal, and what that could point to is a common what? Ancestor. A common ancestor, since we're all using the same genetic code. There are some exceptions, and I wrote them in your notes there for you. So now let's talk about that process of transcription, okay? And we had a little thing to remember it, so let's go right here. The DNA, we were supercoiled, right? We all know about that, but now here we are available. And you know what? This right here, this whole bit, how that's controlled, that's huge, and that's chapter 13, okay? All right, so be right here. The DNA has to unwind and unzip, helicase, and we have 2 strands. Do we need both? No. So let's put away the lagging strand. And we're not making DNA anymore, we're making RNA. So we better use what? RNA polymerase binds to the promoter. This is huge because sometimes things can bind to the promoter. So when I want to bind, oh, I can't, oh, I can't, oh, I can't. If I can't bind to the promoter, I can't boom boom, right? Sounded bad. Okay, I can't make the mRNA. So this is chapter 13, right here. All about chapter 13. Okay, thank you. Okay, RNA polymerase binds to the promoter. Boom, boom, boom. You make your mRNA, peel it off, set it over there. Go get your lagging strand, zip it back up, set it aside. Get your mRNA. Put on your cap and your tail, and you cut out the introns before it leaves the nucleus. Then you go take this mRNA and go put it on the workbench. What's the workbench? Ribosomes. Every 3 bases are called a codon, and that's complementary to the anticodon. And the anticodon is located on the tRNA, and T stands for? And what is it transferring? Amino acids. tRNA 1 lands, tRNA 2 lands, actually over there. We'll go here. And the two amino acids join together in order to build a protein. Like wings. And then you die. Oh, we could just loop it into that other song, right? Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop by meiosis, and they adapt and die. Yay! Okay. Now, okay, let's watch this little video. Maybe one of these doesn't play well. Yes, today is Friday. Ready?
The synthesis of messenger RNA is called transcription. Transcription begins when RNA polymerase recognizes and binds to the promoter region on the double-stranded DNA molecule. A particular subunit of the messenger RNA, called the sigma factor, participates in recognition of the promoter region. Soon after transcription is initiated, the sigma factor dissociates from the RNA polymerase. RNA polymerase moves along the template strand of the DNA, synthesizing the complementary single-stranded messenger RNA molecule. Synthesis is in the 5' to 3' direction, with new nucleotides being added to the 3' end of the growing messenger RNA molecule. As the RNA polymerase advances along the DNA, it melts a new stretch of DNA and allows the previous stretch to close. When RNA polymerase reaches...
Sorry, I want to pause for a minute. Look at this. Why is this called 5' right here? Because we read the leading, which read from 3 to 5, and we're making 5 to 3, right? If we're reading 3 to 5, the complementary nucleotides, the sugars, the way they're facing, are going to be the other direction, right? Okay.
which is a specific sequence of nucleotides on the DNA called the transcription terminator. A hairpin loop structure forms in the messenger RNA, causing the RNA polymerase and the messenger RNA to dissociate from the DNA.
Yeah, that breaking off part, there's a self-complementary region on the DNA, okay, so that when it's this long chain and it hangs off, it wants to fold back on itself. That helps it to fall off. And there's also a whole bunch of adenines in a row on the DNA, and that also contributes to, this is the end of it, our mRNA is enough, okay? Now, this mRNA, it got longer the farther we moved along here, right? Started out shorter and then it got longer. Yeah? Okay, that's going to come up in a little bit when I ask you a question. Okay. Now, check with your bio buddy. Is this transcription going correctly? Are there any mistakes, or is this a-okay? Go ahead, check with your bio buddy.
It is.
Are we good? We are good. Everything's going to plan, right? You're reading one strand of the DNA, you're reading the leading strand, and then you're making complementary, and the right bases are pairing up. Now, we know, because we've already talked about it, every 3 bases on here is going to be called a what? Codon. And that's going to be complementary to a anticodon on the tRNA, and the T is transferring an amino acid. And if I was going to have an anticodon right here, what would it be? A really? A what? A U, because this is still RNA, right? AU what? G. Okay, who determined this was going to be AUG ultimately? The DNA. Yes?
So the multiple adenines that were in the video that you just talked about?
Yes.
Those would be the stop codon that you said?
Oh, okay, that's, I know, there's so many things to keep track of, okay? Stop codons are located on the mRNA. I mean, they were on the DNA as well, but when we looked at that codon chart, that's to stop bringing tRNAs. That's to stop this. There isn't a tRNA that will land on those. Okay. All right, now, take a look at this picture. The RNA is right, I'm sorry, the DNA is right here. That's the DNA. That's the leading strand of DNA. On here are many, 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 many RNA polymerases. And they are making many, 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 many copies of this DNA. Where is the 3' end? Is it on the left or the right? On the DNA, where is the 3' end? On the left or on the right? Make a claim to your bio buddy, support it with evidence, and give a reason. Okay, where am I going to put each? 3's on the left or on the right?
Right.
Oh, there's some debate. What did I tell you? The longer you've been reading the strand, the longer the RNA is. So where's the shortest? The shortest pieces are here on the left, the longest pieces are down here, yeah? So this would be the 3' end of the DNA, and this would be the 5' end. And as the RNA polymerases are moving along here, okay, so here's an RNA polymerase, and it's moving along here, the longer the mRNA strands are going to be that are made from the DNA. Okay? So ask your bio buddy, sunshine or shadowlands? So if I was making it, if I was right here, then my mRNA strand that I would have made at this point is about this long. But as it moves along here, my mRNA strand is probably this long. And as it moves here, now it's this long. Do you see what I'm saying? Because I'm copying, now I've copied all of this. Where here I'd only copied all of this. And here I'd only copied all of this. Do you see now? Okay, are you all right? Okay. So this is using DNA to make RNA. And here in this case, lots of RNA. Okay, and what is that process called when you use DNA to make RNA? Transcription. Say it. Transcription. When you go from here, be here, here to here, that's transcription. When you go from here to here, that's translation. Okay? Do you want to record this song? This part? Because I do not have that. Okay, here, let me pause this for a minute. Okay, here we go. Now, let's talk about how we make that mature RNA. Take a look up at the top. In purple is your RNA polymerase that bound to the what? Promoter. And it's making the mRNA. You can see the mRNA has both the orange and yellow parts to it. The parts that are yellow are getting removed, and those are called introns. And what we have left behind are exons. They put on a cap right here of 7-methylguanosine in blue, and then 100 to 200 adenines here at the end, and it's leaving the nucleus. Now, once it's left the nucleus, you could have several ribosomes. The mRNA is here, and we said it binds to the ribosome, right, the workbench. Well, this RNA could stay right here, and this ribosome could go and read along it, and then another ribosome could come and read along it, and another ribosome could read along it, and that's called a polyribosome. So it can be translated extensively. Now, how would a prokaryotic cell be different? What would a prokaryotic cell have? It wouldn't have that nuclear envelope. This part wouldn't be here. And they don't have room in their small little cells to carry a bunch of genetic junk, which could be failed attempts at evolution and some other structural components of the linear chromosomes, okay? But as soon as the RNA polymerase is right here and the mRNA comes off, a ribosome can get on it immediately and start reading it. So you could be mid-transcription. You could be like this in transcription. It's starting to come off, and a ribosome could attach onto it because it doesn't have the barrier of that nuclear envelope. Okay, so that would be something that's different. Now, that splicing process is taking place right here, and I want the youngest bio buddy to explain what's going on. List all the things top to bottom. Go. That's funny. It was already uploading the other one. Okay. All right. Now let me talk to you about this. There are reasons why, okay? Valuable reasons here to have introns. And I want you to look on your notes. Go to, we're at 12.4. And underneath the second box, it says RNA molecules undergo processing, number 1. And then number 2, function of introns. Ping pong back and forth with oldest bio buddy going first with A. Go ahead. Okay, so I would pick 3 key words out of there. Why you have introns. The first key word I would say is what? What would be your first key word? I would pick a word that starts with V.
Variability.
Variability. Gives you variability. What's your second keyword? I would start with a C. What does it give you? Control. Right? Control. So I see variability, I see control, and then the third one, I would see another C one. It allows for crossing over. Okay, variability, control, and crossing over. Let's talk about variability first. Okay, your immune system, super, super important. Okay? You are pre-adapted to defend yourself against any pathogen you would find anywhere on this planet. Right? Usually we only become sick once. Okay, as we're building up our immune, haven't learned about the immune system yet, but we will. As you're building up your little soldiers to fight your battles, you might get sick once, but then you get memory cells, memory B and D cells, that allow you next time you get exposed to it. That's why you get vaccines. Expose yourself to a weakened version so you can build up your armies and your memory cells. So if you really get exposed to polio, you can fight it off. Okay, all of you were born being able to fight it. Okay. Every flu that comes around, you were born being able to fight it. Okay? That all came from just 3 genes spliced differently. That's what gives your millions of variety of both B and T cells from just 3 genes getting spliced differently. Think of your mRNA as thousands and thousands and thousands of letters, okay, in a long line. I could write different poems with that one long line of letters depending on which letters I eliminated, right? Are you with me on that? If I eliminate the right ones, I could make the letters only T, H, and E out of there. And so the first thing I would see is the, right? And then I could eliminate the next one, and the next ones I would see, so the next word I only see is big. Okay, so depending on which parts it's the same series of letters, but depending on what I rule out and what I cross off, depends on what I will then read from that, what protein I will get out of it. Okay, so that's the variability part. What's the second part? Control aspect. Okay, RNA is single-stranded, so when you have mRNA and you want it to be read and it goes out and gets on the workbench, what's the workbench? Ribosomes. If a small RNA, it could be a small interfering RNA, okay, could go out there and bind and all of a sudden make a section of it not single-stranded but double-stranded, you can't, there's no place for tRNA to land because there's RNA there making it double-stranded, okay? So that could control in good ways and in bad ways, right? And then what's your third one? Crossing over, which really lends it to the first one, which is what? Variability. Those are areas where your chiasmata, where you can do your crossing over. Okay, so youngest bio buddy, tell them the benefits of those introns, please, in your own words. Go ahead. Use this in your explanation. All right, so moving forward, this is I'm not sure if this exact picture is in your new book or your old book. I mean, I know it's in your old one, but I'm not sure if it's in your new version of it. But you can see you have you're transcribing, the DNA is here in blue, and then your primary transcript, and then your mature mRNA. You have your cap and your tail, and your spliceosomes are the ones that are going to be removing it. So you could have variability in your spliceosomes, which look like the green eggs on there, that are removing different pieces of that primary transcript. Okay, now another picture. Take a look right here. Okay, so here we have in our red, we have our DNA. The RNA polymerase is what color? Orange. And then the messenger RNA is what color? What color is the messenger RNA in this picture? Yellow. Good. And the spliceosomes are what color? Green. Okay, and they're splicing it into different combinations. And I don't think this link is working anymore. Let me check. Oh, it is working. It didn't work in last lesson. Okay, here we go.
Most eukaryotic genes are composed of numerous short coding sequences called exons, interspersed between long stretches of non-coding sequences called introns. The initial messenger RNA molecule copied from a gene includes introns as well as exons. Before this RNA molecule can be used to make a protein, the introns must be removed by a process called RNA splicing. A structure called a spliceosome plays a key role in RNA splicing. The spliceosome is composed of several small molecules called snRNPs, or SNRPs. SNRPs are made up of protein and small RNA molecules. Formation of the spliceosome begins when one kind of SNRP binds to the 5' end of an intron, and a different kind binds to the 3' end of an intron. Additional SNRPs then interact with the complex, bringing the two ends of the intron together and causing the intron to form a loop.
So do you see where it binds determines which part is cut out, right? And that's how you can control that, where it binds.
This also gathers all of the SNRPs together as the completed spliceosome.
SNRPs are happiest when together.
The intron is then cut out and the exons are joined or spliced together. The spliceosome then falls apart as the SNRPs are released.
All right. Now, I'm so glad that worked. Let's do our first question. Are you ready? It's villains. 777 number. Oh, whoops. Sorry.
Wait, that's the.
Oh, I skipped a question, I think. I'll go back to it. Some of you are really in the video. It uploaded, so we're good now. Why didn't you tell me? Poor Miss Litton, not paying attention. Help me. Good job, the Riddler.
I bet you can't guess who that is.
Is it you? Okay, I am very sad because it looks like 3 of you are struggling and you need a shame bell rung right near you. Which type of RNA carries the amino acids? Who's the mule? Who carries the amino? tRNA. Okay? One of you put mRNA. The M stands for message. Where did it get the message from? DNA. And 2 of you put rRNA. rRNA becomes part of the ribosome, and that is the workbench. Look at your Bio Buddies clicker right now. Hold them accountable. Do they get a gong or a bell of shame?
Why does
Sorry, little struggle, little struggle. I'll fix it eventually. Turn you board. Oh, there we go. Okay. Let's try for at least 1 hundy, yeah? 3, 2, Dr. Doom, Jafar, you're killing me. Will this be a hundy? Wait, hang on, here we go. Will it? No, no, it is not a hundy. Good job, the Riddler. All right. Okay, so let's deal with B. The central dogma of molecular biology states that DNA is a template for only DNA replication. No. Yes, all your RNA DNA serves as a template for that too. So no to B. Woe to the person who chose B. States that translation precedes. What does precedes mean? Does translation come before transcription? No, because you're translating what got transcribed. You have to do this before you do this. Yes? Transcription, then translation. And then E, all of the above, no. So which means that they thought D was right too. Pertains only to prokaryotes because humans are unique? No. It's what? And it points to a common ancestor. Check your BioBuddies clicker. Gong or little bell of shame? Clarifying questions. Like, no, I can't because you ring the shame bell on me. Anybody though? Now's the time. It's safe. In Tuesday, it won't be safe. Right? Right now, it's safe.
It's a safe zone.
Safe zone. Come on, guys. This could be your hundy.
That was way before you. Don't even try. Doesn't matter. That means I was first.
Sorry.
Stop.
Easy. Guys, this should be super easy. Stop fiddling around. I think I've got like 4th graders. Confusing me. Five, 4, 3, 2. Dr. Doom.
Oh my God.
This is me, shocked. Okay. mRNA synthesis is called, some of you said translation. What are you synthesizing in translation? Proteins. Proteins. Okay? When you make RNA, it is called what? Transcription. This. Transcription. This is translation. Check with your BioBuddy right now because you should not be missing this at this point. Check with your BioBuddy.
No, I put D in.
Oh, but it marked you wrong?
Yeah.
Oh, okay.
Yes.
We did that one. It's a map. It's a map. Guys, do you want me to stop the video before we go into translation so we break it up, transcription, translation? Yes? Okay.
Yeah.
All right, let's start talking through it while we're waiting for these few poor people to finish. Okay, are we done? Okay, let's see. The Riddler. All right, let's talk through it. All right, yeah, every single one of you understood A, so that's good. Everybody understood A. Did everybody get B? Yes. Did everybody get C? No. So let's talk about C. The enzyme RNA polymerase synthesizes RNA. Is that an understanding, or is that just kind of a fact? Fact. Okay. But does it make sense to you that you would use RNA polymerase to make RNA? Because you use DNA polymerase to make DNA. So this is also true. Okay. Yes, somebody picked D. Let's talk about D. Spliceosomes remove the exons and splice together the introns. How do we make this correct? Spliceosomes remove the introns and splice together the exons. Okay? See if your bio buddy is one who picked D. And ring that bell. Did everybody pick? Oh, yes, somebody did. Okay. RNA is made in the cytoplasm of eukaryotic cells. No. Where is RNA made? In the nucleus. Okay, of eukaryotic cells. A cap, did everybody get F? No. A cap is put on the 5' end of the primary transcript. Yes, because that primary transcript you read from 3 to 5, making 5 to 3, and the cap goes on first, right? And then the tail. Yeah, because tail's in the back. Okay. Check with your bio buddies, see if they were part of the 66% or no. All right. Before I move on to translation, did it go weird again? Okay. I'm going to go ahead and stop this video and not it, pass or play on giving the details of transcription. And if you're watching this really late at night, you should have a piece of toast.